Thank you for the invitation to speak. I somehow wasn't on the mailing list until now, so I'm also looking forward to watching future talks. So as Alex mentioned, my interests lie largely in Earth's oxygenation history. And I've more recently started thinking about how that informs our ability to recognize exoplanet life, and specifically the utility of oxygen as a potential biosignature. So this talk is going to begin on Earth, and we'll spend the majority of our time here on Earth. But then towards the end, we'll zoom out and consider exoplanets a bit. Okay, so first and foremost, the early Earth was essentially entirely devoid of oxygen. And this is, it sounds like a bad thing from our human perspective. We require lots and lots of oxygen, but this is actually a really good thing for Earth's habitability. And that's because even though there is a lot that we don't quite understand about the origin of life here on Earth, everything that we think we know about prebiotic chemistry is just wildly incompatible with the presence of oxygen. And so the fact that early Earth was anoxic is ultimately the first step towards evolving a diverse biosphere here on Earth. Of course, we now have 21% oxygen in our atmosphere by volume, and that oxygen is the consequence of oxygenic photosynthesis. But as we'll talk about, the relationship between biological oxygen production and atmospheric oxygen accumulation is really quite complicated. And so that's going to be the focus of this talk. But we're interested in understanding these dynamics, not just because oxygen on an exoplanet might signal the presence of photosynthetic life there, but also because oxygen is ultimately a strict requirement of other types of life. So all of our cute pets, this is my dog, this is my cat, require oxygen just like us. And so if we want to understand the potential—
Sorry, Stephanie, I think I just realized that you've been trying to change slides, but they haven't been changing.
Oh, I have been changing slides on my screen.
Okay, yeah, so I was wondering, I don't know what's going on.
I can stop and reshare. Let's see.
So it's a second screen, right? There's what we can see at the bottom left of the current screen, there's arrows you can be clicking to change the slide, maybe, or I don't know if you needed to do it there or.
Here, I'll start from scratch. So
okay, sure.
Okay, so you should hopefully be seeing my normal PowerPoint window.
Yeah.
Yeah, okay. Oh, I see what's happening. Okay.
Okay, good. I'm glad one of us does.
No, I understand. My Zoom panel is hiding the controls here. But now do you see my cute animals?
We do, but you have to swap displays.
All right, that is easy. I can do that.
Great, we see them.
All right, wonderful. Okay, so as I was saying, if you want to understand the potential distribution of adorable cats and dogs in the universe, you need to understand how Earth became oxygenated and the potential for other worlds to become oxygenated as well. Okay, so I've already alluded to this being a complicated history. One of the major challenges here is that oxygenic photosynthesis did not immediately oxygenate the atmosphere. And so here I'm showing the history of oxygen in Earth's atmosphere in the bottom panel, with time advancing from left to right, compared against milestones in the history of life here in the top panel. And so you can see here that oxygenic photosynthesis evolved around perhaps 3 billion years ago, but didn't rise during the Great Oxidation Event, the GOE, until about 2.4 billion years. And so this is a significant time lag. All right, so how do we know that oxygenic photosynthesis predates the GOE by so much? Well, evidence comes from several different archives, but one of my favorite is isotopically light molybdenum in the Pongola supergroup. So here I'm showing molybdenum isotope composition here on the x-axis against the iron-manganese ratio here on the y-axis. And so because manganese here is in the bottom, manganese increases going downward on this axis. And so what you can see here is that with increasing manganese content, you get progressively lighter and lighter molybdenum isotopes. And so this is suggestive of oxygen because manganese oxides preferentially absorb isotopically light molybdenum from seawater. And so the incorporation of light molybdenum into these rocks implies that the manganese was originally in the form of manganese oxides, which we think require oxygen to form. But again, this is just one of many lines of evidence for biological oxygen production in advance of the GOE. So here's several more. So we've got evidence from molybdenum isotopes, uranium, nitrogen cycling, et cetera. And these data span a huge range of time, beginning about 3 billion years ago and advancing all the way up to the GOE itself. Okay, but at the same time, we of course have really compelling evidence for an anoxic atmosphere, despite this evidence of oxidative processes. So my favorite is the preservation of detrital pyrite. So this is pyrite that's been weathered and transported long enough distances to become rounded, but apparently in low oxygen conditions, because otherwise it would have been oxidized instead of transported. More recently, the smoking gun for an anoxic Archean atmosphere is considered to be these sulfur isotopes. And so this is kind of a complicated record, but the take home here is that prior to the GOE, we have lots of variability and high magnitude mass independent fractionation of sulfur isotopes. And then following the GOE, it collapses and disappears, never to return again. And so because this is a signal that we think is produced within anoxic atmospheres through photochemistry that's inhibited in the presence of an ozone layer, this again strongly suggests that we have an anoxic atmosphere during the Archean. Okay, so how do we reconcile evidence for oxygen accumulation within seawater with an anoxic atmosphere? Well, the solution is that oxygen was initially accumulating within marine oxygen oases. And the way this works is that cyanobacteria in the ocean are able to make oxygen very quickly and transform their local environment on a timescale of hours. Likewise, oxygen in a broadly anoxic atmosphere is incredibly reactive. And so atmospheric chemistry can destroy oxygen, again, on a timescale of hours. But the process of sea-air gas exchange is actually very slow. So this is a process that equilibrates the surface with the overlying atmosphere on a timescale of about a month. And so because life is making oxygen so quickly and any oxygen that escapes the atmosphere is consumed so quickly, this slow communication between the two reservoirs allows oxygen to accumulate within the surface layers of the ocean. And so as Alex mentioned, many years ago, I did a master's with Lee Kump, and we sought to quantify to what extent, what oxygen concentrations and to what aerial extent oxygenation within the surface ocean was possible. And so here I am showing a map of dissolved oxygen concentrations in surface seawater on the left, and then on the right is a cross-section of the top 140 meters of the ocean from the South Pole to the North Pole. And so what you can see here is that we're predicting like single-digit micromolar dissolved oxygen over broad swaths of the Archean surface ocean. These oxygen concentrations are certainly low compared to modern, which is typically in the ballpark of 250 to 300 micromole. But these are actually biologically significant oxygen concentrations. And again, I'll note that it's restricted to the surface layers where oxygen is actively being produced, and it is not accumulating at depth in the ocean. Okay, so this is the same data now presented a little bit differently in a histogram instead of a map. And so what you can see is that although about half the surface ocean had very low or no oxygen, half of the ocean had micromolar levels of oxygen, and about 15% of the ocean had dissolved oxygen concentrations in excess of a couple micromole per kilogram. And this is important because aerobic respiration by microbial ecosystems requires only nanomolar levels of oxygen. So what this means is that the majority of the Archean surface ocean may have been hospitable towards aerobic microbial life in the Archean. And even at the higher end here, some of these oxygen levels are actually suitable for simple animals, so things like sponges. Although I'm not arguing that sponges existed in the Archean, to be clear. Okay, so in addition to oxygen oases, there are also hints of what are called whiffs of atmospheric oxygen in Archean sediments. So the most famous example comes from the 2.5 billion year old Mount McRae shale. And within that shale, we see a systematic enrichment in molybdenum. And this enrichment in molybdenum implies that there was a significant amount of molybdenum dissolved in the ocean. And that's important because molybdenum is primarily supplied to the ocean through oxidative weathering of the continents. And so that you get high level molybdenum enrichment in Archean shales implies that there was oxidative processes occurring on land. And so one interpretation of that is that there was actually oxygen within the atmosphere prior to the GOE. Okay, but what exactly are whiffs is really poorly understood. So one scenario is that some people think of whiffs as plumes of oxygen that are emanating from these oxygen oases within surface seawater, maybe a similar phenomenon within lakes. And this, is appealing because this is simple and it's mechanistically linked to oases, and so we don't need to invent an entirely new phenomenon to explain whiffs. The problem, however, is that we don't actually know, what oxygen levels we would expect within plumes, and if that oxygen would be long-lived enough to actually meaningfully interact with the continental crust. And so plumes almost certainly existed in some form, but whether they are capable of generating these signals is less clear. A second scenario for whiffs is that they actually represent kind of failed oxygenation events in which the global atmosphere became transiently oxygenated. And so maybe there was some kind of oscillatory oxygen behavior in advance of the GOE. And this is something that I'll talk about at length in a couple of slides. The third scenario is that perhaps whiffs are actually totally unrelated to atmospheric oxygen at all. Maybe whiffs are simply the expression of microbial communities on land that are actually producing oxygen directly within the weathering environment, oxidizing and mobilizing molybdenum, but not actually oxygenating the atmosphere. And so this is appealing in that it simplifies things. We again don't need to invent a new mechanism to explain whiffs. But if this is where the molybdenum is coming from, one obvious question is, why are molybdenum enrichments transient within Archean sediments? Why don't we see a relatively continuous record of molybdenum in Archean rocks? Okay, so now I'm going to walk through each of these three scenarios and and see to what extent they are reasonable explanations of whiffs. Okay, so to do this, I actually updated that model that I used way back during my master's and revisited this idea of Archean oxygen oases. And so among the changes that I have made, there are two that are most important. So first, now I am calculating oxygen in the atmosphere rather than imposing it. So a decade ago, what we did is we fixed oxygen levels to be low in the atmosphere and said, can oxygen accumulate in the surface ocean? And the answer to that was a resounding yes, it definitely can. Now we're starting with an anoxic atmosphere and an Archean-like biosphere and seeing to what extent we expect oxygen to actually accumulate in the atmosphere. And it can actually vary both in space and time as a result of that. And the other thing that we're doing that we didn't a decade ago is that we are actually now characterizing seasonality and time variability within the marine environment. Okay, so first and foremost, we got a qualitatively identical result. We again simulated oxygen oases within the surface ocean. And the distribution of oxygen is spatially similar, and the concentrations are also spatially similar. This pleased me because many of the simplifying assumptions that I made a decade ago turned out to actually be reasonable things to have done. But we did learn a lot in doing so. So, for example, we learned that oxygen oases are a summer phenomenon, and so oxygen is restricted to exclusively the summer hemisphere. So here we have northern hemisphere summer on the left, northern hemisphere winter on the right. And while oxygen concentrations approach 10 micromole per kilogram in the northern hemisphere summer, those waters become essentially anoxic during the winter months. And so this is potentially an explanation for, I've already mentioned that these oxygen concentrations are enough to support simple animals like sponges, but if this is a seasonal phenomenon, perhaps that's why we don't see more complicated life at this time. Okay, then I've already mentioned that we actually calculated atmospheric oxygen now rather than imposing it. And so we did find that these oases in the ocean are actually associated with plumes of oxygen in the atmosphere. And they reach concentrations of about 1 ppm. So this is approaching what you need to oxidize pyrite, but not significantly exceeding that threshold. But the key here is that these plumes are really tightly coupled to oases, both in space and in time. And that's because the lifetime of oxygen in these plumes is actually about 3 hours. And so these plumes are not wafting over the continents. They're not interacting with rocks. And they're probably not mobilizing molybdenum. I know I lost my cursor. There we go. Okay, so I don't think that plumes of oxygen emanating from marine oxygen oases are a viable explanation for so-called whiffs of oxygen in the Archean. But what about transient oxygenation of the global atmosphere? Well, so you can drive changes in the global atmosphere by either increasing the source of oxygen to the atmosphere or decreasing the sinks of oxygen to the atmosphere. And so the sources could include things like, if you increased organic carbon burial or pyrite burial or hydrogen escape, you might increase oxygen on a global scale. Or at the same time, if you decreased the oxygen sink associated with oxidation of volcanic gases, for example, you might also drive an increase in atmospheric oxygen. And so in this really simple framework, whiffs might actually just be explained as a natural expression of variability as your sink and your source are of similar magnitude. And so on the left, I'm showing a scenario where you have a slowly increasing biological source that has some natural variability superimposed on that. And as it nears the threshold, the oxygenation threshold, or exceeds the sink for oxygen, it might rise above and fall below that threshold several times before permanently staying above. And so that's a simple version of a whiff. The opposite scenario would be a waning volcanic sink for oxygen that also has some variability superimposed on it, such that the sink might fall below the source a couple of times before permanently staying below. And so either of these could drive pulses of oxygenation followed by deoxygenation. Another way in which you could get rising and falling oxygen is if there's some kind of negative feedback on oxygen, in which oxygenation disfavors further oxygenation. And so one possibility here is that perhaps increases in oxygen somehow drive decreases in nutrient availability, such that more oxygen inhibits the biosphere from making more oxygen. And so such a feedback might exist within the nitrogen cycle. So on Archean Earth, where the ocean is essentially globally anoxic, the stable form of nitrogen is ammonium. Today, our ocean is well oxygenated and nitrogen exists as nitrate. But in a redox stratified world in which you have oxygenated surface waters overlying anoxic deep waters, you can get oxidation of ammonium to nitrate. But if that nitrate is mixed into the anoxic deep ocean, it's not reduced back to ammonium, it's actually reduced to dinitrogen, which is not biologically available. And so there's kind of a bit of a bottleneck here, and where increasing oxygen might actually drive nitrogen loss from the system, reduce primary productivity, reduce oxygen production, and tend to result in declining oxygen. And so this is a sort of feedback that could explain oscillatory oxygen dynamics in advance of the GOE. Okay, so no to plumes, maybe to transient oxygenation of the global atmosphere. Now, what about those terrestrial photosynthesizers and mobilization of molybdenum from the weathering environment that way? Well, so I was initially skeptical of this idea, but actually I think I have some data that actually supports it now. And so I'm warming up to this possibility. So in support of the possibility that microbial communities on land are actually driving whiffs, I'll note that the Mount McRae Shale isn't the only Archean molybdenum enrichment. And so we actually documented a systematic molybdenum enrichment here in the 2.7 billion year old Roy Hill Shale of the Drina Formation. So it has similar shape to what we see in the McRae, but it's an order of magnitude lower than the famous whiff. And so it's qualitatively different in that way. Now within the Roy Hill Shale, we see that molybdenum here on the y-axis scales with increasing organic content here on the left x-axis. And so this is expected in modern sediments today. Molybdenum, which is soluble as molybdate in seawater, is converted to molybdenum tetrathiomolybdate, which is reactive towards organics and forms complexes. And so in euxinic environments, we see higher molybdenum concentrations with increasing TOC. Of interest here, though, is that there's this abrupt break in the slope, in the relationship between molybdenum and TOC here. And so using conventional wisdom, the implication here is that this reflects a decrease in bottom water molybdenum concentrations. And so that's certainly possible. But when we look at molybdenum against sulfur, we see no such break in the slope. And so what we argued is that actually molybdenum uptake into sediments wasn't limited by bottom water molybdenum concentrations and their conversion to tetrathiomolybdate and interaction with organic carbon. It was actually limited by the amount of sulfide in the system. And so we were variably sampling the amount of molybdenum in seawater because there was incomplete conversion of molybdenum to tetrathiomolybdate. And so the scenario we proposed was that this is actually controlled by pulses of sulfur delivery to the ocean, perhaps in the form of volcanic sulfur, and that there was some background steady flux of molybdenum from the continents to seawater. And then we just variably sample it depending on how much sulfide accumulates in seawater and the fraction of the molybdate pool that's ultimately converted to tetrathiomolybdate and incorporated into marine sediments. And so this would be consistent with terrestrial photosynthesis and ongoing mobilization of molybdenum from the continents. Okay, so putting everything I've said so far together, we have some evidence for marine oases beginning by 3 billion years ago. At 2.7 billion years ago, we start to see perhaps some hints of terrestrial photosynthesis and oxygen influencing weathering processes. And then by 2.5 billion years ago, the Mount McRae whiff looks very different than everything that came before it. And this might be evidence of some kind of oscillatory oxygen in advance of the GOE, either due to just natural variability or perhaps due to feedbacks on oxygen. But then even after the GOE, even after oxygen rose, it seems that oxygen oases likely persisted, perhaps throughout the entire boron billion. And the reason that we think this is actually quite simple. So here I am showing on the right a map of surface, of modeled surface oxygen concentrations under Proterozoic conditions. And you'll see that this is remarkably similar to all of the maps I've been showing you so far for Archean oxygen concentrations. And basically the idea is that the surface ocean in the Archean was supersaturated with respect to oxygen, to the extent that even after the GOE, the surface ocean was still supersaturated with respect to atmospheric oxygen. And so the composition of surface seawaters actually probably didn't change dramatically in response to the changing atmospheric composition. Now, fast forwarding now to the Ediacaran, we actually see a different type of oxygen oasis. So in Ediacaran sediments, we see evidence for extensive microbial mats at the seafloor. We see this in the form of textured surfaces on sedimentary rocks. And these would have been photosynthetic communities that were producing oxygen. And it's possible that the earliest animals, the Ediacaran fauna, were actually exploiting this mat environment as sort of a benthic oxygen oasis. So for example, on the right here, I'm showing some of the earliest burrows. This is a trace fossil called Helminthoidic nides, and it tracks the movement of an early bilaterian through the oxygenated surface layers of a microbial mat. And then we have some much larger guys, like here, this is Kimberella, who's scratching at the surface of the mat. Now this is potentially a feeding strategy or potentially liberating oxygen bubbles from the underlying mat. And so again, early animals may have occupied oxygen oases, and oxygen oases, even though we think of them as an Archean phenomenon, are sort of relevant for the majority of Earth's history. Okay, so now let's zoom out a little bit. I've promised to pitch this in terms of exoplanet life detection. So what does this long history of oxygen oases on Earth mean for atmospheric oxygen as an exoplanet biosignature? Okay, so I've shown you this oxygen history already, but now I'd like to point out a couple of issues. So first, oxygenic photosynthesis evolved quite early, by 3 billion years ago, but that means that there's a period of time before that of greater duration than the entire Phanerozoic with very high oxygen levels, for which Earth's biosphere was entirely anaerobic. And so there are Earth-like biospheres that have absolutely no oxygen at all. Then I've spent the majority of this talk talking about this interval of time here, where oxygenic photosynthesis is active and ongoing, but is somehow not oxygenating the atmosphere. And again, this period of time is of similar duration to the well-oxygenated Phanerozoic. And then there's an even bigger problem, and that is that there is potentially as much of it as a 2.5 billion year delay between the origin of oxygenic photosynthesis and the achievement of oxygen levels that would be, remotely detectable by an alien civilization with our foreseeable technologies. So now I've added to this oxygen history a hypothetical detection threshold with current and proposed space telescopes. This is the sort of oxygen levels we would require to see on another planet. And so, again, a 2.5 billion year delay between biological oxygen production and the achievement of kind of remotely detectable oxygen levels. So in terms of oxygen as a biosignature, it seemed that oxygen is actually quite an unreliable biosignature because atmospheric oxygen levels are not a passive reflection of surface environments. In particular, non-detection of atmospheric oxygen does not preclude biological oxygen production, oxygen accumulation, and even aerobic microbial ecosystems within the ocean. In the case of the Ediacaran fauna, which we're exploiting a benthic oxygen oasis, lack of observable oxygen might not even preclude animal-grade complexity. And just more generally, it's clear that Earth-like planets probably don't actually have remotely detectable oxygen just based on the history of our planet, where we've had remotely detectable oxygen levels for less than 10% of our history. Considering Earth's future evolution helps a little bit, but oxygen in our atmosphere will eventually begin to decline in about 1 billion years when the silicate weathering feedback draws down CO2 to low enough levels that photosynthesis becomes challenging and oxygen production shuts down. And so even considering the future evolution of Earth's atmospheric oxygen is present at detectable levels for a minority of Earth's history, a very small fraction ultimately. Okay, so more generally, because we don't know, we've made a lot of progress in terms of characterizing by when and by how much oxygen rose in our atmosphere, but the why has really remained challenging so far. And because we don't know why oxygen rose when it did, we don't yet know if atmospheric oxygen was an inevitable or even likely outcome of photosynthesis on Earth. And so the extent to which we should expect to leverage atmospheric oxygen as a biosignature on other planets really depends on resolving this why question. Was it a fluke that it took so long for oxygen to build up in our atmosphere? Might we expect other planets to oxygenate sooner in their histories? So there are several explanations for why it took so long to oxygenate Earth's atmosphere. But a favorite hypothesis recently has been that Earth's early ocean had a scarcity of nutrients, so primary productivity and oxygen production was low. So in other words, life has been making oxygen here on Earth for a very long time. It just wasn't very good at it because there were limited nutrients around. Another possibility is that nutrient recycling was just simply less efficient. So on Earth today, one of the essential ingredients for nutrient recycling is ocean upwelling. And that's because the biological pump exports nutrients to depth. But then on global average, the ocean is actually stably stratified with respect to density. And so vertical mixing is generally inhibited. We have warm sunlit waters sitting on top of very cold, salty, dense bottom waters. And upwelling, which brings back nutrients that have been lost to depth, only occurs in very specific environments, most famously off the coasts of continents, where winds are pushing offshore flow, and that requires that water rises up from below to replace it because the continent itself is obstructing return flow at the surface. And so this results in surface divergence and upwelling. And so if we look at a map of chlorophyll concentrations in Earth's ocean today, where green and red are high levels of chlorophyll, blue is very low levels of chlorophyll, we see that photosynthesis is overwhelmingly concentrated at high latitudes where that density stratification breaks down, along the coasts of continents where upwelling is occurring, and to a lesser extent along the equator where surface divergence due to opposing directions of Coriolis deflection also enables upwelling to occur. And so I wondered, how might upwelling and nutrient recycling have changed through Earth's history? And to what extent might that affect biospheric productivity, oxygen production, and the oxygenation trajectory of our atmosphere? So I used a GCM called Rocky 3D to characterize upwelling under a variety of circumstances, including rotation rate. So here I'm showing upwelling normalized to present day Earth upwelling for Earth-like planets with differing rotation rates. So I varied rotation rate such that day length varied from 10 days down to 12 hours. And so what you can see here is that upwelling drops off with increasing rotation rate. And this is actually a kind of conceptually intuitive relationship. And that's because Ekman transport, which is the wind-driven circulation of the ocean, the potential for surface divergence, scales proportional to the wind forcing divided by the Coriolis parameter, which on global average is just simply the rotation rate. So qualitatively, what that means is that the wind-driven circulation of the ocean and upwelling from divergence resulting from that depends on both the wind and rotational effects. And the faster and faster the planet rotates, the less effective the winds come at driving divergence. And so planets that rotate slower than Earth the winds are more capable of driving upwelling, whereas planets that rotate much faster than Earth, the rotational effects become increasingly important and the winds are less effective at driving upwelling. Okay. I also looked at the relationship between ocean upwelling and surface pressure. And I found that ocean upwelling increases with increasing surface pressure. And that's because the wind stress, the wind forcing that drives surface divergence, scales proportional to surface pressure. And so the more an atmosphere is pushing down on its underlying ocean, the more effective the winds can drive divergence in the surface layers and promote upwelling from below. And again, that upwelling brings with it nutrients, potentially greater biospheric productivity and oxygen production in particular. And so I mentioned this because Archean Earth, we think, rotated faster than today. And so here I'm showing the same oxygen trajectory that I've shown many times here in the top panel. And the history of Earth's day length in the bottom panel. And so day length increases through time, perhaps being as short as 12 hours way back in the Hadean, and it's of course now 24 hours today. And there are some striking similarities between this, this history of day length and Earth's oxygenation history. And in particular, it's possible that maybe this boring billion corresponded with resonance stabilization of Earth's day length at about 21 hours. And the second increase in oxygen here, the neoproterozoic oxygenation, apparently coincides with the resumed slowing of Earth's rotation rate. And so we've argued that there might be some kind of mechanistic relationship between those two, specifically a long-term increase in ocean upwelling, nutrient recycling, and biospheric oxygen production. Likewise, surface pressure has potentially changed through time as well. And so characterizing ancient surface pressure is, of course, challenging, but here are two examples of attempts to do so. So here we have raindrop craters, and here we have vesicles in basalts, so basically ancient bubbles. And so the size of the bubbles and the size of the impact of raindrops are both related to surface pressure and the extent to which surface pressure is pushing down on the lava in this example. And so these records suggest that Archean surface pressure may have been much lower than today. And so putting this all together, our Archean world rotated very fast. It potentially had low surface pressure and would have had limited coastline to stimulate coastal upwelling. And so maybe the Archean biosphere was much less productive than today because nutrient recycling through upwelling was much less effective. And then through time, as Earth's rotation rate has slowed and surface pressure has increased and coastline to support upwelling has increased, perhaps there's been a long-term increase in nutrient recycling, biospheric productivity, and biological oxygen production that maps to the oxygenation of our atmosphere. Okay, so I'm running on time and I have covered a lot of ground, and so I will pause briefly to summarize. So I hope that I've convinced you that oxygenic photosynthesis dramatically predates atmospheric oxygenation here on Earth. And the manifestation of this is oxygen oases within the surface ocean and whiffs of atmospheric oxygen potentially. But these are two distinct phenomena. Oases don't imply whiffs. In the context of exoplanet life detection, this complicated oxygen history really renders oxygen an unreliable biosignature, to the extent that non-detection of atmospheric oxygen on another planet doesn't preclude biological oxygen production or even the potential for animal-grade complexity, given that the Ediacaran fauna existed in a microenvironment that was supersaturated with oxygen with respect to the broader water column, which was itself supersaturated with oxygen with respect to the overlying atmosphere. And because we don't know why oxygen rose when it did, we don't know if it was a likely or inevitable outcome of oxygenic photosynthesis on Earth, or whether there might be planets that might be more likely to support oxygen requiring complex, intelligent life. But our simulation suggests that maybe slower rotation and higher surface pressure are two things that might promote oxygenation. And it just so happens that Earth's rotation has slowed through time and its surface pressure has potentially increased through time, which together might result in a significant increase in biospheric oxygen production here. Okay, so thank you for your attention, and I will accept questions.
Okay, great job, Stephanie. Thank you very much. This is where we're going to transition to our question and discussion section. And as we normally do, anybody may ask questions either by typing a question into the chat, so I can ask it for you, or you can raise your hand, or you can type the star into the chat, and I'll try to get everybody in the order that we're hearing them.
So maybe I can start with two questions. Stephanie, great talk. So first question, like assuming that in Archean we don't have glacial deposits, and it was also argued that size of emerged continental crust, at least early on, could have been much smaller. How do you think it would affect upwelling rate? Because you are not producing sort of thermohaline circulation in high latitudes, and you also missing continents. So that's first question. And second, how well is changes in rotation rate established? I presume it's coming from tidal deposits, maybe, or maybe theoretical. And what's the basis that in middle Proterozoic we have no change in rotation rate?
Those are two great questions. I'll start with the first. So you're right that the absence of significant landmass in the Archean could potentially severely affect ocean upwelling. So we've done experiments. The ones that I showed today, of course, had modern continents, but we've also done aquaplanet simulations. So the other end, remember, no continents at all, and we've done hypothetical supercontinent scenarios as well. And we have found that the modern continent configuration is by far the superior situation for ocean upwelling. That's because we have lots of north-south coastline along which upwelling can occur. And the aquaplanet scenario has the least upwelling. There is still equatorial upwelling that results from the opposing directions of Coriolis deflection on either side of the equator. So there's divergence that occurs along the equator regardless of continentality that could support some nutrient recycling. And then one clarification that I'll make is that ocean upwelling is largely a wind-driven phenomenon, and this is somewhat distinct from the thermohaline circulation. So it's true that what goes down must come up, and so strengthening of thermohaline circulation will strengthen return of nutrients from depth. But what we were characterizing was largely a wind-driven process that operates independently of that. Oh, and your second question. So rotation rate, in the relatively recent past, I think there are some sedimentary archives that can be leveraged to estimate rotation rate, but going all the way back to the Archean, there are essentially no physical constraints on rotation rate. And so that is almost entirely a theoretical thing. And it has to do with the evolution of the moon's orbit, which is slowly retreating from Earth. And the idea that it was stabilized for so long during the middle Proterozoic comes from, again, models that predict some sort of resonance with the atmosphere. The physics escape me, but it's a model result that has been found by multiple groups at this point.
Thank you.
Okay, thanks. We have a couple questions in the chat and another hand raised. So we'll start with Daryl Long asks, can you use molybdenum levels to estimate terrestrial biomass?
Ooh, I think that would be really hard to do. So the sequence of events, what you would want to do is you would want to translate sedimentary accumulation of molybdenum into seawater accumulation or seawater concentrations, and then from seawater concentration infer a flux from the continents. And that's hard to do because, I mean, we don't even have good constraints on the volume of the Archean Ocean. So I don't know. Theoretically, yes, but I don't know how to do that math at the moment.
Thank you.
All right. So the next question is from Sarah Giles, who says, great talk. Do you think those oases you talk about could have implications for carbon isotope anomalies in the Proterozoic?
I think absolutely yes. Oxygen oases could affect spatial patterns in carbon isotopes because these would be localized environments of methane oxidation potentially. And so you could get both lateral and vertical carbon isotope gradients arising from that.
All right. Jinjin Liu has a question. So Jinjin, you can go ahead.
Sure. Hi, Stephanie. Good to see you on Zoom. I have two questions. One is I'm a little bit confused about when you're talking about O2 supersaturations. I'm just curious because in modern day, the O2 in the surface ocean is 280 micromole, and it's largely a result of both the O2 against respiration and O2 against ALC gas exchange. So I wonder, because we are doing a simple calculation showing that when you lower the PO2, the O2 recycling within the ocean due to respirations is going to increase as well. So I wonder to what extent your supersaturation, sorry, let me rephrase it. So is your supersaturation concept just strictly in the sense of Henry's law coefficient, Henry's law equilibrium, or does it take into account of respirations?
All of our experiments include microbial respiration, yes. But the supersaturation under Archean conditions is largely driven just by the kinetics of sear gas exchange being so slow. But it's respiration that's, if you recall, oxygen levels went to effectively zero outside of upwelling regions in Archean simulations, and that's the consequence of respiration.
Okay, yeah, we can talk about later, but another question is the molybdenum figures you shows on the Archean shell. I just wonder whether you whether that data shows to a slope already, because seems for me, if I recall correctly, that curves, you can still kind of point it out. There's a different slope down there, which is kind of the reason why I'm saying that is because, well, Lee had a paper earlier saying most of volcanism is subaerial. So SO2 should be reduced to to H2S in the process, making the reduction possible. And maybe the molybdenum content was lower is to explain why the slope doesn't go all the way to the higher molybdenum content in the modern environment.
Was there a question embedded in there?
Yeah, just would you mind to go back to the to the data?
Oop, too far. This one.
Yeah, this one.
Mm-hmm.
Like the orange below the curve. I just want to know what you make the conclusion that there is only one slope instead of two.
Oh, sure. So in TOC space, there are definitely two slopes, but in sulfur space, these data do not systematically separate. They're effectively overlapping here, and there's a single slope. And And so in modern sediments, molybdenum uptake is related to TOC. So molybdenum TOC ratios reflect bottom water molybdenum. And that's a kind of conventional molybdenum wisdom. But here, molybdenum is scaling more predictably with sulfur. And so we argued that molybdenum uptake wasn't limited by bottom water molybdate. It was limited by the conversion of molybdate to tetrathiomolybdate, which is the particle reactive species that's actually interacting with organic carbon. And so this sulfur was sort of acting as an intermediate limitation on molybdenum accumulation in marine sediments. And so as you suggested, Lee suggested that there was enhanced subaerial volcanism at this time, and I think that's a fantastic source to have episodic pulses of sulfide into the marine environment, and so episodic enrichment of molybdenum in marine sediments.
Yeah, thanks. That makes much better sense. Thank you.
All right, there's a couple other questions now in the chat. Joanne Bowden asks, in your opinion, were planktonic marine oxygenic phototrophs required to create the GOE? Could terrestrial and benthic marine phototrophs in coastlines oxygenate the atmosphere instead?
I mean, theoretically, benthic communities could make lots and lots of oxygen. But in my opinion, yes, I think planktonic communities were important. And that's because putting in oxygen at the bottom of the ocean is not, there's a lot that oxygen must survive before it can ultimately enter the atmosphere and affect atmospheric chemistry. And so I think it's just much easier to do if you have widespread planktonic oxygenic photosynthesis occurring. That, and so if continental area was much reduced, there's kind of an upper limit on the amount of oxygen that a terrestrial community or even the area of benthic photosynthetic area within the ocean itself would be limited to. So I think you just do more with planktonic bugs.
All right. She says, okay, thanks. Now, Catherine Giles says, thanks, Stephanie, for such an interesting talk. There's many new studies showing significant phosphorus nutrient cycling via windblown dust, which might also follow from the limited land mass in the Archean. Do you think this could have been part of the equation?
Absolutely. So that long-term changes in the total amount of phosphate in the ocean could affect biospheric productivity and oxygenation. I don't wish to debate that at all. I think there is absolutely a role for long-term changes in total phosphate. But I think also that this upwelling and phosphate recycling kind of acts in addition and can amplify those sort of changes.
Okay, great. At the moment, we don't have any more questions or hands raised, or questions in the chat or hand raised. So if anybody wants to go ahead and ask one, feel free to unmute yourself and get involved. If not, we will. Okay, Masha Prokopenko, can you comment on the more recent events of increased upwelling, Miocene, and its influence of atmospheric oxygen?
So there have been some fluctuations in atmospheric oxygen during the Phanerozoic, most famously the Carboniferous. Oxygen dynamics during the Miocene are well beyond my expertise, but presumably possible. I guess the major difference being that once vegetation is well established on the continents, that's playing a very large role in atmospheric oxygen. Whereas this upwelling could be a really important phenomenon in the distant past, where terrestrial communities were more limited. And so I think it's possible, but I don't know how quantitatively significant.
Okay, she says thanks.
So we
I'll give it another second.
nicely end Stephanie's talk on nutrients, which leads to next week presentation by Mike Kipp on phosphorus.
Right, yeah. Right, phosphorus, the phosphorus cycle, yeah. Okay, well, I haven't seen anybody drop anything in the chat, and nobody's stepped up to speak. So thank you so much, Stephanie, for your presentation today. It was very well delivered and very, very thought-provoking, and generated some good discussion. So it's great to have you.
Yeah, looking forward to being in the audience moving forward.
Yeah, and since you're kind of new to us, we do post everything on YouTube. So any past presentation you might want to see, you can check our YouTube channel. It's all there. So with that, everybody else also feel free to go look at the YouTube channel. And we will see you all next week, I guess.
And as in case with Stephanie, password around about the seminars, like we're trying to incorporate everyone. So if we didn't reach to people, password.
All right. All right, everybody. See you next week. Stay safe.
Bye.
Bye.